Thank you very much for inviting me to give this talk and actually talk about the impact of transcriptomics of our understanding of pulmonary fibrosis. Since its introduction in early 2000, genome-scale transcriptome profiling caused an inversion in IPF research to human-based hypothesis. So it allowed investigators to move from animal-based to human-based hypothesis, very important. Transcriptomic studies led to identification of novel biomarkers, therapeutic targets, and mechanistic insights. Transcriptomics, as matured, it allowed for system biology approach to investigate spatiotemporal lung development and microenvironments. And in my talk today, I'm going to address all of these three approaches in more details. As transcriptomics evolved, cohort size increased. So initially we analyzed 10 samples. Today we analyzed hundreds of samples. Transcriptome coverage increased from analysis of hundreds to thousands to 10,000s of genes, from analysis of coding RNA to analysis of non-coding RNAs. Novel insights such as role of MMPs, epithelial activity, development pathways, role of epithelial apoptosis, microRNAs, lung macrophages, mitochondria, and metabolism, and presently usage of single cell to define novel cell types actually came into the focus of IPF research thanks to transcriptomics. Biomarker discovery and really understanding disease progression, acute exacerbations, while analyzing lung, blood, or bile came into the focus of IPF research thanks to transcriptomics. Transcriptomics also allowed for a class comparison amongst diseases, IPF versus other ILDs, and understanding really what differentiates IPF from other diseases. Now, in early 2000, my mentor, Naftali Kaminsky, pioneered the usage of microarrays to analyze human IPF lung. And at that time, a dramatical increase in expression of MMPs, especially MMP7, was actually defined to be increased in IPF epithelium. And in addition to matrix metalloproteinases, all the molecules listed in this table actually are differentially expressed in IPF epithelium. So really usage of transcriptomics in early stage kind of induced a shift in how we understand IPF, from moving from solely really investigating fibroblasts to paying attention to IPF epithelium. As transcriptomics emerged, activation of aberrant development pathways was probably one of the most fascinating findings happen in IPF research thanks to transcriptomics. Amongst these pathways, Wnt pathway candidates such as Wnt1, 3, 5, 7, 10 are actually defined to be increased in IPF lung and studied further. Fibroblasts isolated from human IPF lungs were defined using transcriptomics, and candidates such as FKPB10 or insulin-like growth factor binding proteins defined to be increased in IPF lung. Further, apoptotic response also was characterized using transcriptomics. And TWIST1, CXL12, just some of the candidates that are characterized to be increased in IPF lung thanks to transcriptomics. Now, recently, host defense and role of host defense was defined, and some of the candidates such as defensins 3 and 4 defined as increased in lung, and Agar defined as a decreased in IPF lung. And mitochondria-related genes that are suggestive of dysfunctional mitochondria, actually, and candidate that's probably the most studied, PINK1, was identified as a decreased in IPF lung. Now, DIO2 was identified as an increase in IPF lung, and it was suggestive of perturbed thyroid hormone in IPF. In early stage transcriptomics, lung affected with IPF were compared to lung affected with HP, and investigators wanted to understand really what differentiates these two types of diseases. And T-cell activation, inflammation, immune cell response were actually associated with HP, but not with IPF. Stroma, surfactant, and heat shock proteins were associated with rapid, but not stable progression of disease. Epithelial injury and proliferation associated with acute exacerbation, cilia-related genes associated with UIP patterns in IPF lung, And recently, novel autoimmune endotypes were identified to be commonly shared between IPF and COPD patients and were suggestive of increase in p53 and hypoxia pathway. Now, can transcriptomics help us identify new drug targets and drug development? So initially, DIO2 gene was identified, thanks to transcriptomics, to be dramatically increased in human IPF lung. This gene encodes for the enzyme that converts inactive T4 into active T3 form in thyroid hormone pathway. Now, as we look at this heat map, and we see that what is marked in red is really overall characteristic and specific gene changes that differentiate IPF from other ILDs and COPDs, and we look more into the pathways that are perturbed, what we see is that there is an overall decrease in thyroid signaling and mitophagy. Now, without going into the details of particular molecules here, investigators, Goin Jung and in the lab of Naftali Kaminsky, actually decided to try thyroid hormone as a potential drug for IPF. And in bleomycin-induced fibrosis, actually they tried a treatment with nintedanib, pirfenidone, and in parallel aerosolized T3. And what was observed is that T3 decreases or resolves fibrosis in a more efficient manner than two FDA-approved drugs, which put these thyroid hormone mimetics as really interesting future candidates for drug development. So second drug candidate is miR-29, Mimicry. Approximately 10% of microRNAs are significantly changed in IPF lungs. We know that members of miR-30, LAT7, miR-29 are actually decreased in IPF lungs, in lung cancer, and stimulation with TGF-beta. On the other hand, we know that miR-29 has an inhibitory effect on extracellular matrix proteins and growth factors. So the idea here was to supplement what is missing, okay, to supplement fibrotic lungs with miR-29 and see does it resolve fibrosis. And in this study led by Rusty Montgomery, actually, in bleomycin-induced fibrosis, miR-29 mimicry resolved that fibrosis. And presently, this drug is in development with Miragen Therapeutics. Now, transcriptomics also helped us prioritize protein biomarkers. MMP7, osteopontin, COMP, CXCL13, and CCL8, well-studied proteins, biomarkers in lung, in serum, in plasma, in BAL. And out of all of these proteins, MMP7 is probably the most validated biomarker, and it's predictive of increased mortality in multiple cohorts of IPF patients. Transcriptome also allows us, when peripheral blood was analyzed, to actually reveal 52 gene outcome predictive signature. Now, this is very interesting, is that this signature allows to divide patient populations into two groups, high and low risk. And this signature is actually validated in 6 cohorts in 425 participants, so truly amazing. We see that in patients with high risk, with poor survival, there is a decrease in CD4 and CD28 T cells and increased in activated monocytes. Presently, this signature is in further development in Kaminsky's lab under leadership of Rami Unteram, and he's using a single-cell approach to really understand better how each of the gene of this 52-gene signature changes in PBMC cells. So we see that downregulation of the genes is associated with B, T, and NK cells, while upregulation, what is marked in green here, is associated with monocytes and with actually poor survival. Another very interesting findings here using single-cell transcriptomics is identification of actually Treg cells in PBMCs that are correlated with progressive fibrosis. We also see them increased in IPF lung, and they correlate with actually poor outcome. Now, 52 gene signature, what is the future perspective of this signature? So we know when gender, age, and physiology index was added to this 52 gene signature, it significantly improved its mortality predictive accuracy. This signature may outperform biomarkers solely used, such as MMP7 or postin or CXL13. It may also outperform changes in circulating cells, fibrocytes, gene polymorphism, or aging biomarkers such as telomere length and mitochondrial DNA. However, this signature as such is not ready to enter into the clinic. We need more work on this signature. We need to validate it and to validate it in a longitudinal prospective study to establish universal reference value to determine specificity and association with disease progression and acute exacerbation. But this signature is really a very interesting candidate for future development. Now, BAL gene signature is also indicative of IPF outcome. And here in a study led by Antje Prase, a set of genes was identified in BAL and as well as in basal cells present in a BAL. And as we can see, the signature is actually validated in numerous cohorts and definitely increase prediction accuracy of outcome of IPF patients. Now, what is really interesting here is that investigators were, by identifying the strong signal of minority cells in PBMCs, were intrigued actually to see what's going on in IPF lung, and they identified the presence of these airway basal cells in the close proximity of fibroblast foci. So a very interesting finding thanks to actually identifying the signature in BAL, and Antje Prase will talk more about this today. As transcriptomics matured, it really allowed for system biology approach, and we performed analysis of differentially affected microenvironments in the IPF lung. So we know that we use CT as a diagnostic technology to identify presence of traction bronchiectasis and honeycomb cyst in IPF lung. However, in research, we use micro-CT, and micro-CT has simply a deeper resolution. It really allows investigators to kind of observe the changes in IPF tissue similar at the level of histology. In this particular study, led by John McDonough, investigators sampled the IPF lung from apex to base, from less fibrotic to more fibrotic, and as we can see here on the right side, IPF1 stands for the areas of the IPF lung that is non-fibrotic, actually normal-looking lung. IPF2 stands for mild fibrosis, and IPF3 for advanced fibrosis. When investigators performed gene differential analysis, they identified the presence of these core IPF genes that are actually perturbed in IPF1, but as well as IPF2, IPF3, which means that we have actually perturbation in the gene level even in normal-looking IPF lung. Further on, investigators used more complex data analysis approach and identified distinct gene expression tracks. Now, this is also very exciting. It allows us to see what is up, what is down-regulated in IPF lung if we go from IPF1 to IPF3 stage of fibrosis. And let's say on a figure B, A, and B, we see that extracellular pathways actually are increased in IPF1, while figure C and D is suggestive of immune response pathways increased in IPF2 stage, and so on. Now, investigators came up with the first dynamic regulatory model of transcriptional change in IPF lung using this approach. So we can see that activity level of fibrotic gene expression increases in early stage and it stays up as disease progresses from early to late. MIR29 is identified as an early stage regulator and LET7 as a late stage regulator. We also see steady increase in activity level in genes correlated with structural fibrosis and honeycombing. When these data sets were actually analyzed in parallel to single cell data, it allows us to see what of the cells are enriched in early increased pathways, and those were the fibroblasts, actually. In advanced increased progressive pathways are actually enriched for B cells, basal cells, ciliated cells, and macrophages, and early decreased pathways are enriched for 81 and 82 cells as well as macrophages. Now, this approach really allows us to understand now at the level of cell populations where is the change, how change happened, and what pathways are perturbed. So next, what about single cell resolution? So we all know that presently this new technology came into the transcriptomics research, and presently we have more than 500,000 cells analyzed, more than 100 human lung tissue analyzed, more than 40 IPF tissue analyzed. So these tissues are analyzed by the groups cited in these slides, 5 groups, and the first phenomenon that came actually was described using the single cell was the bronchialization. Actually, the presence of these epithelial cells in the distal lung that acquire a typical mixed differentiation state, and they are present in alveolar epithelium, but actually looks like airway epithelium. Next, rare cells were identified using the transcriptomics, and they're defined as aberrant basaloid cells that are localized in the proximity of fibroblast foci. Senescent score was identified to be highest in alveolar type 2 cells, specifically isolated from fibrotic lungs. Ectopic peribronchial endothelial cells were identified at the edge of fibroblast foci and in the areas of bronchialization. IPF-specific archetypes amongst fibroblast, myofibroblast exist, and Naftali Kaminsky was talking about that yesterday in more details. Profibrotic macrophages subpopulation overexpressing SPP1 that localize in the lower lobes of fibrotic lungs was identified. So really using this single cell, it's really exciting moment for us in IPF research to really understand better what's going on in IPF at the cellular level. And I would like to invite you all to visit IPF Cell Atlas. You can use this QR code for easy access, and you can mine these data sets further. Now, towards the end of my talk, instead of summarizing this review, I would like just to provide a few directions where we are actually and where we should be. So over the 20 years, we really gathered a lot of knowledge so that we know what kind of study questions we can answer with transcriptomics. We can use different kind of tissues, lung, bowel, blood. We can go from bulk to sorted cells to single cells. We can use arrays, RNA-seq on single-cell RNA-seq. Now, we have available bulk-seq analysis, and actually a lot of data sets is accumulated using this approach. Bulk analysis is a well-defined analysis, well-defined protocols, doesn't have a good resolution, okay, but the gene signal is very strong. Next step is the microenvironments. So that allows us to identify or to analyze sorted cells or cell population. So it has a better resolution, protocols are optimized, we can use it in fixed, in frozen, in fresh tissue, and also it gives us this kind of a spatial context of gene expression in IPF lung. And presently we have a single-cell technology which is in development, protocols are still in development, and sometimes tissue dissociation actually can differentiate between protocols. This approach provides a high resolution, but it's also very costly. Now, today, to do transcriptomic studies, the suggestion is really to take in consideration all of these three approaches, to take in consideration data sets that are already publicly available, to really help us answer any question of interest. And as a set of recommendations towards the end of my talk, for foundations, I would really like to suggest that you continue sponsorship of transcriptomics-based projects, and especially young investigators. These projects and techniques are truly exciting, but also very costly. For advocacy groups, please continue supporting usage of transcriptomics for biomarker and drug discovery. And for patients, I would really like to thank you for participating in research, because we do all this for you, but we cannot do it without you. With this, I would like to conclude my talk, and thank you for your attention.